So welcome to the Genetics 311 theme park. Before a theme park can actually become a theme park, it starts out as a sequence full of grass and trees, which is like our given DNA sequence up here in black. However, once it gets transcribed into mRNA, which is equivalent to the blueprints being made, our new 5' to 3' mRNA strand is coded, which you can see in red, and now our theme park can be built/translated into amino acids, which down here are the specific amino acids that code for our mRNA strand. A nonsense mutation occurs when a single nucleotide is substituted and changed so that it reads as a stop codon instead of a regular amino acid. The typical stop codon is placed at the end of an mRNA sequence. However, if placed at any other point, then the amino acid would prematurely terminate as the correct protein would not be produced. This substitution, as you can see here, we went from UGC to UGA, resulted in a transversion pyrimidine to purine as the nucleotide base A was substituted for C, changing the amino acid sequence from cis to a stop codon. So our cis becomes the gates, our stop as you leave. Missense mutation occurs when one nucleotide is substituted and alters the existing codon by changing into a completely different one, meaning that the whole 3 base codon codes for a completely different amino acid. So in this case, we changed the nucleotide G to U, which changed the amino acid completely from ALA to SIR. So in this case, our ALA now is SIR, which changes the sequence of which roller coasters are ridden at the theme park. This substitution resulted in the transversion purine to pyrimidine. A silent mutation occurs when a single nucleotide is substituted for a different base yet codes for the same amino acid, and so the protein will not be affected. In this case, we went from nucleotide base C to U, however, it still codes for the amino acid ASN the same. In this case, our roller coaster ASN will still remain the same even though a new loop is added to it. A read-through mutation occurs when a stop codon is changed to a codon that codes for an amino acid, which results in a longer protein. UGA was changed to UUA as the nucleotide base G was substituted for U. This changed the stop codon to read into the regular amino acid LU, which as you can see resulted in a longer protein as there is no longer a stop codon in the sequence. So our stop codon, the gates at the exit, is now LU, PRO, GLY as follows. This substitution resulted in a transversion purine to pyrimidine.